In this video, we're going to go through an introduction to the study of tissues, which we call histology, right? Just looking at section tissues under a microscope. And as an introduction, we're going to go through epithelial tissues, which is one of the four major categories of tissue types that you typically see in an anatomy and physiology class. Right, so the general concept here is, remember, tissues are collections of cells that have similar shapes and functions. If you combine tissues together, you get entire organs. So when you put a sample under a microscope and start looking at it, you might be seeing an individual tissue, or you might be seeing an organ that contains multiple tissues. And we sort those tissues out into four major categories, right? Nervous tissue, epithelial, muscle, and connective. And we're going to have videos going through all of those different types. For right now, we're going to focus on epithelial tissues, and we'll get to others in a bit. But just remember, these are categories. So under each major category, there are different types of tissues, and you find those different types of tissue in sometimes many different organs. Especially in the epithelial and connective categories, you find those tissues across pretty much every organ system throughout the entire body. And so the goal is we're learning to recognize patterns and connect the structure of these tissues to their function, because they're not shaped the way that they are randomly, right? They have a purpose behind it. And the idea is to put these things under a microscope, look close at their cellular structure, and see if we can connect that to how they function the way that they do. All right, so the thing that you'll see connecting all of these different epithelial tissue types together is that they are all the lining of something. Like the definition of an epithelial tissue is one that lines an open space or a cavity. So think the lining of your GI tract, right? Stomach, intestines, the lining of the airway and the lungs, lining of blood vessels. Technically, even the outermost layer of skin, the epidermis, is an epithelial tissue because it sort of lines the exterior of the entire body. And then the rest of the universe would be the open space that it's connecting to. Right? So to start talking about epithelial tissues, we got to give them names. And the naming system is pretty convenient because it's just described based on the shape of the cells and the number of cells in the tissue. All right, so these two terms, simple and stratified, are telling you how many layers there are. A simple tissue, epithelial tissue, really just means that there's a single layer of cells, and a stratified tissue just means that there are multiple layers of cells stacked on top of each other. And the easy way that you're going to tell these apart when you put them under the scope is look at nuclei. Remember, every cell, almost every cell in a human, only has one nucleus. And so if you see multiple nuclei piled on top of each other, even if you can't always see the border of individual cells, you know that there have to be multiple layers as opposed to a single. All right. The second component, right, on this side of the table is the shape of those individual cells. Squamous refers to these thin, flat, sort of pancake-shaped cells. Cuboidal refers to squarish, cube-shaped cells. You're not seeing them in 3 dimensions under a microscope because everything is flattened, but you can sort of visualize or imagine that they would be shaped like cubes, like building blocks, if you pulled them out. And columnar, that are shaped like relatively tall, skinny rectangles. Think columns like, columns in a building, pillars that are holding up a structure, right? So put the terms together. Simple cuboidal, one layer of cube-shaped cells, stratified columnar, multiple layers of rectangular column-shaped cells. And there are a couple exceptions to the rule. One of them, this term pseudo-stratified. Remember, the prefix pseudo always means fake or false. And so pseudo-stratified because the nuclei are offset from each other, and it can give the false impression that they're stratified, right? It'll make it look like there are multiple layers, sort of trick your eyes into thinking that there are, but it's really just one layer of cells that are kind of twisted around each other, and the nuclei are a bit offset. So they still fit into the columnar category, but we call them pseudo-stratified because of that appearance, right? So as we go through these different tissue types, we're going to look at a lot of these major categories, give examples of each. Just remember, it's not exhaustive. There are hundreds of these tissue types throughout the body if you get very picky about naming them, and we're just going to do some common examples of each. All right, so the first one, simple squamous. Simple is telling you there's a single layer of cells. and squamous is telling you that they're very flat and thin, right? So the example of this that I'm using, this particular tissue came from the alveoli in the lungs, and so all of this space out here would be the actual open airway, and in an epithelial tissue, the open space that the tissue surrounds is always called the lumen. Right? That term is used all throughout anatomy. Lumen just means open space. So the inside of your stomach is a lumen. The inside of your airway in the lungs is a lumen. The inside of an organelle in a single cell can be called a lumen. Right? So in this picture, you can roughly see the shape of individual cells because when we look at this tissue, these dark purple ovals that you see here, the darkest stain on here is a DNA stain. So those are the nuclei of cells. So you can just roughly outline individual cells by looking at where a single nucleus is. And you see that there's only one cell thickness across this barrier. So it's simple. The cells are relatively flat, and so they're squamous. The follow-up question to that is why? So that's the anatomy part of it. The physiology aspect is, remember, the entire purpose of this airway is to get oxygen into the blood vessels that are running through this tissue. Right? The capillaries here are way too small to see because they've been flattened when the tissue was prepared. But the goal is we want to get O2 in and CO2 out. That's the whole purpose of the lungs. And the thinner that membrane is, the better off you are. The downside of that is the epithelial layer is very delicate. It's easily damaged, easily infected. That's why inhaling dust or debris is damaging to the lungs, and it's why the lungs are easily infected by things like viruses or bacterial infections and pneumonia, is because you don't really have a whole lot of physical defense there. All right, so in all of anatomy and physiology, get used to talking about trade-offs, right? What are the costs? What are the benefits? In this example, the cost is vulnerability, easily damaged. The benefit is you get gas exchange and you can't live without oxygen. So there's really no choice but to accept all of those downsides. All right. Okay, simple cuboidal epithelium. Same concept. This is showing you a section that came from a kidney. And the kidney is really just a network of tubes that move fluid through a set of filters to determine what molecules go into your urine and what are returned back into the blood. And in these tissues, it's never going to be perfect, but if you start drawing shapes around each of these cells where you can see a nucleus, you see that they are roughly cube-shaped. Again, it's biology, not math, so it's not going to be perfect. It's not like they form perfect right angles, but they're roughly cuboidal. And there's a single layer because those cells that I just drew around belong to this particular lumen. And this set of cells belongs to this lumen. This set of cells belongs to this lumen. And so every open space has one ring of cells around it. So we say that they're simple, right? The function of these cells is some sort of absorption or secretion. These cells in the kidney are determining what molecules, and water for that matter, get absorbed back into the blood and which ones go into the urine, right? And so if things stay trapped in the lumen, that's going to be shipped out to the bladder, eliminated as urine. If molecules get pulled out the opposite side, there are blood vessels in here that you can't see that will absorb those nutrients and fluids back into the body. All right. Okay. Next one, simple columnar. The example that I'm using for this came from the GI tract. This happens to be a section of small intestine, and this is your open white space. So you know that's going to be your lumen, and it's a little bit difficult to see here, but notice the shape of these sort of long, stretched out, oval-shaped nuclei. These are very, very tall columnar cells, right? So each one of these structures that I'm highlighting is a single columnar epithelial cell. And this is another good place to point something out because it's shown so clearly. Epithelial cells are what we call polarized cells. You may have heard the term polar in chemistry, which means a molecule is positively charged on one side and negative on the other. In cells, polarity just means something about the shape of that cell is different on one side than the other. So this surface up here, the side facing the lumen, we say is the apical surface of those cells. This surface down here on the underside, we say is the basal surface of that cell. Sometimes people call this the basement membrane. That's what's anchoring you to the underlying connective tissue, which is this tissue under here. So the difference is, notice that the apical surface of these cells up here looks just a little bit fuzzy. That fuzzy border is actually a bunch of structures called microvilli, which we'll see a more clear picture on the next slide. Those microvilli are responsible for absorption of nutrients right out of the small intestine across these epithelial cells. And then the basal side of the cell is exporting them into the blood vessels underneath that are in this connective tissue. Okay, so we say that they're polarized because this side, the apical side, looks different and functions differently than the basal side. Right, the apical surface is supposed to be pulling nutrients into the cell. The basal side ships those nutrients out of the cell into the underlying blood vessels. Okay, so pay attention to that. Every epithelial cell has an apical and basal side, right? Like back here, this would be the apical side of these cells, and this would be the basal side underneath. So if you want to figure it out, always just look for the lumen, right? Look where the open white space is. Whatever side of the cell is directly facing the lumen, that has to be the apical surface. Okay, so if we want to zoom in a little bit closer on the simple columnar tissue, this is an image that was taken by an electron microscope, which can show much, much higher resolution than a light microscope can. And so those microvilli that we talked about, this is what they look like in fine detail. They're really just extensions off of the cell membrane that weave up and down and up and down. And the entire purpose of it is we're trying to maximize surface area, because the goal of this tissue is to absorb nutrients out of the GI tract. And anytime you're absorbing nutrients, they pretty much have to go through some sort of transporter. All right, so if you remember in a past class probably, if my black line represents a cell membrane and these little transporters that I'm drawing, think of these as an active transport pump. If you want nutrients, right, pretend that this is, let's say that my little black dots represent glucose or sodium or any nutrient that you're pulling in from the GI tract. If you want those nutrients to enter the epithelial cell, right, this is the inside of the cell over here, you need more of those transporters. So instead of having a flat membrane like this, if we have microvilli, that increased surface area gives you the ability to plug many, many, many more transporters into the membrane, right? And more transporters means faster diffusion. So best estimates by people that study this histology are that if you took a person's small intestine and laid it out perfectly flat, so you flattened all the villi, you flattened these microvilli, make the membrane like a sheet, it would be approaching 1,000 square feet of space, which is absolutely massive. And so you get many, many, many more times the membrane surface area with this method than you would if the tissue was just flat. All right. Okay, another columnar epithelial tissue, but this is that pseudostratified variety that we mentioned. And again, they are columnar, but they look like they're stratified because the nuclei are offset from each other. And notice on the apical surface, which is over here, right, this tissue happened to come from the trachea, right, part of the respiratory tract. So this would be the lumen over here, which is just the airway. So this would be where the air breathing in and out of the lungs would be passing. And these structures that you see here that sort of look like fuzz, those are cilia. Cilia are similar to microvilli in that they increase surface area, but they're not there for absorption. They're there for movement. The respiratory tract secretes a bunch of mucus, and cilia push that mucus up and out of the airway so that it turns around and goes down the esophagus, because the stomach, which is what the esophagus leads to, has a bunch of acid. Acid can kill bacteria and viruses that are contaminating the lungs. And so as soon as you see those cilia, that's a pretty good giveaway that you're looking at a pseudostratified epithelium in this case, and look for those offset nuclei as another clue. Okay. All right, next type. An example of a stratified epithelium. Stratified, remember, just means multiple layers. So these cells, this is your epithelium, this is the basal side. All of these nuclei are individual cells, so you can clearly see many layers, 5, 6, 7, maybe even 10 cell layers thick. This sample happened to come from the epidermis in the skin. So this up here would technically be your lumen, but it's not really a lumen, that's just the outside world, right? That's the exterior of your skin. And as these cells divide and move up this way, all of this wavy stuff is keratin protein. That's what we mean by these cells being keratinized, right? And so there's a progression of these cells at the bottom dividing, moving up, dying, and basically becoming packed keratin that eventually sloughs off and has to be replaced over time. All right, so in this case, this is almost a perfect opposite of the lungs. Right? In the lungs, we wanted a very thin, delicate, squamous epithelium. Here, we want a thick, multi-layered epithelium because we care about physical protection way more than absorption or secretion, because in general, you don't want your epidermis secreting a whole lot of anything other than maybe some oil, right, to waterproof the surface of the skin. So again, think about those trade-offs, make a comparison, ask yourself, why is this tissue structured the way that it is? All right, next one, transitional. So in this slide, it's a little bit hard to see, but this is your dividing line between epithelium out here, and this is connective tissue on the inside. We call this transitional epithelium, and it's pretty much exclusively found in the urinary system, like the ureters and the bladder, because when the bladder fills, it has to stretch, right? And so when you have a full bladder, these epithelial cells tend to look like this, where they're sort of piled on top of each other, and they're balled up. They look a little bit more cuboidal in shape, or round, like they're globular. And as the bladder stretches and fills even fuller, they'll be stretched out like this. I should have probably called this one partially full. And the fuller and fuller it gets, the more these cells will stretch out and become flat. If they were completely empty, right, you would expect to see cells that are very, very tall and skinny, right, in the other direction, right, sort of bunched up on each other. And so the more and more you fill the bladder, the more those cells stretch and flatten out because the bladder is filling like a balloon, and you need those cells to stretch out to accommodate it. So technically, transitional falls into the category of stratified because there all are multiple layers. But since they change shape, we give it that name of being transitional because the shape of the cell transitions. Okay, a few other quick loose ends to tie up. This idea of intercellular junctions isn't unique to epithelial cells, but it's relevant here because we've got to talk about it at some point. Intercellular junctions are really just proteins that link cells to each other. So if you think about epithelial cells, if they weren't physically connected to each other in some way, the cells would just fall apart, right, into a pile of loose individual cells. And so the idea is we can use these proteins, right, like tight junctions or gap junctions or anchoring junctions to physically connect cells together and make sure they don't fall apart. The benefit of tight junctions is they get the name tight because they form a tight seal that is effectively waterproof, right? You find these in the lining of the GI tract so that you can be selective about what molecules get absorbed by your intestines and which things stay trapped in the GI tract to be excreted. So think of this as almost like a zipper that very, very tightly seals cells together. Gap junctions get their name because this would be cell number 1, and this would be cell number 2 over here. We're linking their membranes together. Gap junctions have a literal gap, right? An opening or a pore between them. So very small ions and molecules can diffuse between cells, right? You find these gap junctions, the easiest example is in the heart, because when the heart contracts, it's an electrical signal. You let ions diffuse between cells to make sure that all the cells of the heart contract at the same time. So it lets cells communicate with each other. All right. And anchoring junctions, we're just going to summarize, right? These different types, desmosomes, adherens junctions, and hemidesmosomes, are different categories of anchoring junction. The main idea is these are here strictly for physical support and connection. So as an example, these would be what are used to hook the basal side of epithelial cells into the underlying connective tissue to make sure that they don't detach and fall apart. Okay, and last thing, we go over exocrine glands here briefly, just because the lining of exocrine glands by definition are made of epithelial tissues, because exocrine glands secrete into surfaces or open spaces. And remember, that's the definition of an epithelial tissue, right? They line open cavities and spaces. And so when you think exocrine, think sweat, saliva, oil or sebum under the surface of the skin. Anytime you're secreting like a bulk product. This is different than endocrine, which is hormones that are secreted into the blood, like insulin or growth hormone, for example. So we split them into categories based on what they secrete. Merocrine glands secrete liquid only. Sweat or saliva are good examples. Apocrine glands secrete more than liquid. They actually break fragments off of their cells, and the cell fragment becomes the secretion. A mammary gland is a good example because it makes sense. Milk is made out of lipids and proteins and water from cells. That's what the cytoplasm of cells are made of. And so we just break whole chunks of the cell off and then it regrows to replace what was lost. Holocrine glands, hollow, think whole. The whole cell ruptures and dies to become the secretion. So the entire cell is secreted, which gives you a lot of lipids. Sebaceous glands, which are the glands that secrete the oil in your skin, are an easy example of that. The consequence is those holocrine cells, they have to have stem cells underneath that are constantly dividing to replace the ones that die, because the whole cell dies and breaks off to become that secretion. All right, hopefully this was helpful. In the next video, we'll move on to connective tissues. Make sure that you flag the playlist and subscribe to the channel in case anything new pops up. That way you can see all these videos in order and get a progression to cover the entire section.